Hello, this is Amanda Welch welcoming you to this Bite-Size Bio web seminar, which today is sponsored by BioLegend. BioLegend develops and manufactures highly recognized world-class antibodies and reagents at an outstanding value. Our product portfolio includes tools for research in immunology, neuroscience, cell biology, cancer, and stem cells. We offer more than 18,000 products and collectively are cited in over 27,000 peer-reviewed publications. BioLegends is headquartered in San Diego, California, and operates under an ISO 1345-2003 certified quality management system. Today's presentation is titled Simultaneous Proteomics and Transcriptomics, the Future of Single-Cell Analysis, and is being presented by Dr. Marlon Stoeckius, a senior research scientist in technology and innovation at New York Genome Center. Marlon received his PhD in 2011 from the Systems Biology Department at Max Delbruck Center in Berlin, Germany. He went on to do a postdoc at the Berlin Institute for Medical Systems Biology, where he studied the transcriptome and proteome of early C. elegans embryogenesis. He continued his studies of embryogenesis at his postdoc at Yale in 2013. And from there, in 2015, he became a research scientist at the New York Genome Center, where he studies single-cell multiomics. As always, we'll have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen. I'll put them to Marlon at the end. The recording of the webinar will be available at bit.ly/single-cell-analysis. That's bit.ly/single-cell-analysis, all one word, lowercase. So now over to you, Marlon, for the presentation.
Thank you, Amanda, and good afternoon, everybody. First of all, I'd like to thank, of course, Bite-Size Bio and BioLegend for giving me the opportunity to present our work here today. My name is Marlon Stoeckius. I'm a senior research scientist in the Technology Innovation Lab at the Genome Center. Before I start, I would like to note that the work presented here is free from oversight from BioLegend, although BioLegend is the exclusive licensee for a patent application related to this work. Today I will talk about two technologies that we recently developed at the New York Genome Center. I will first talk about CiteSeq, which is a method that allows you to measure the immunophenotype and the transcriptome from single cells. I'll first introduce the method and then also talk about a few updates. And then I'll talk about cell hashing, which is a method that allows you to multiplex and superload many samples in one single-cell RNA sequencing run. The scale of droplet-based single-cell RNA sequencing has really transformed single-cell genomics in the last, let's say, 2 years. Now you can really measure the gene expression in tens of thousands of cells in parallel, and there are even data sets out there which go to more than 1 million cells. And here's an example from a collaboration with the lab of Rahul Satija, where we profiled the gene expression in 20,000 cells from synovial fluid in rheumatoid arthritis patients. And the first thing that you then do with these type of data sets is you cluster cells based on their gene expression profiles, and then you visualize it in these so-called t-SNE plots. And every dot here in this plot is a cell, and when these little islands form, that means these cells are more similar to each other. And then you can look into the different gene expressions in the clusters and look for marker genes for cells that you know or cell types that you know, and give these different clusters names. And then you can easily see that our sample from the synovial fluid was infiltrated by a lot of immune cells, but then there were also fibroblasts in the sample. And of course, it's really easy to distinguish now transcriptomically the plasma cells and B cells and all the fibroblasts from T cells. But if you look closely into the T cell cluster, you see that we really only pick up 3 different types of T cell clusters, and it's just barely CD4 and CD8 T cells. However, we know there are much more different types of T cells than just CD4 and CD8. We can't even distinguish memory and naive here that easily in this data set. And that's because these cell types are transcriptionally very similar, and it's very hard to pick up the differences in single-cell RNA sequencing. So on the other hand, we have flow cytometry and CyTOF, which has been used for many years to really characterize deeply different immune subsets in samples based on cell surface markers. And here there's one example from a relatively easy gating strategy from using only 7 markers, which then allows you to distinguish relatively easily CD4 and CD8, different CD4 and CD8 subsets within the sample. So you can clearly see the naive from effector memory, central memory. You can also pick up the different NK cell subsets. And these methods have been used for a while now to characterize the immune system. However, these methods have clear limitations compared to single-cell RNA sequencing. Firstly, they're inherently biased because you only measure markers for proteins or cell surface markers that you know, for which you have antibodies also. And secondly, the data is relatively limited to the number of markers that you can multiplex. Because for flow cytometry, for example, you can routinely measure between 4 to 15 markers, depending what type of flow cytometry you have. And CyTOF, you can go up to around 35. So these are really essentially two totally disconnected worlds or disconnected disciplines. On one hand, we have single-cell RNA sequencing, which really gives you only the RNA readout from many cells in parallel. And on the other hand, you have flow cytometry or CyTOF that gives you the protein readout for a few proteins, but then for millions of cells. So we asked us, how can we merge these two technologies to really get protein and mRNA expression from single cells at a really large scale? So antibodies are used to measure the abundance of a protein in flow cytometry or in CyTOF. And these antibodies are then labeled with fluorophores that are essentially measured. However, we can't measure these fluorophores in sequencing. So we asked us, how can we get a sequential readout from an antibody? And since most of the single-cell RNA sequencing workflows are oligo-DT-based, so they're using oligo-DT primers to reverse transcribe the mRNAs, we asked us, how about we just tag those antibodies with an oligo that mimics an mRNA molecule? So the way those oligos would look like is they would basically have a long poly A tail that mimics an mRNA molecule and would be incorporated into oligo-DT-based single-cell RNA sequencing workflows. And then it would have an antibody barcode, which is essentially the identifier sequence for this one specific antibody. And then, of course, you would have a PCR handle, which would allow you to specifically amplify this product out and make a sequenceable library out of it. And since it's a DNA barcode, you can easily imagine that there are more sequence combinations than fluorophores or heavy metal isotopes that you need for flow cytometry or for CyTOF. So a 12 nucleotide barcode, for example, can easily code more sequences than antibodies that are available for sale. So the way the method then works is you simply stain your cells the same way you stain your cells for flow cytometry or CyTOF, with pools of these DNA barcoded antibodies. You wash away the unbound antibodies, and then you pass those tagged or stained cells through any sort of oligo-DT-based single-cell RNA sequencing workflow. And here I'm simply exemplifying this with dropseq, where every so often a single cell would get encapsulated in a droplet together with a microparticle that brings along the oligo-DT beads for reverse transcription. And then within these droplets, the oligos from the antibodies and the mRNA molecules bind to the oligo-DTs on the beads. And depending on the technology that you're using, you either get the reverse transcription happening in bulk as for dropseq or in the emulsions as for 10x. And then, essentially the mRNA molecules and the antibody oligos will be tagged with the same cell barcode from within this droplet. And since the antibody oligos and the cDNAs have very different size fractions or very different size characteristics, they are separated as part of the standard single-cell RNA sequencing workflow, where you isolate the full-length cDNA after amplification. So the antibody oligos then are essentially amplified, or the antibody library essentially is amplified from a fraction that is discarded as part of the standard workflow, and it doesn't have any alterations or interference with the standard cDNA library preparation for the RNA molecules. So we asked us, what's the ideal model to really test CytSeq? So we then turned to the immune system, which, as I said in the beginning, has been really well characterized based on cell surface markers and also by RNA sequencing. So cell types are essentially well defined based on RNA and protein markers. And importantly, they are very well characterized and high quality antibodies available that we need for this type of assay. So for our proof of principle experiment, we made a panel of 13 CytSeq antibodies to really well characterize immune markers, and then stained cord blood mononuclear cells with this panel and passed them to 10x genomics. And we obtained around 8,000 cells. And the first thing that we did is then cluster those cells based on their gene expression profiles. And here, again, see this visualized in a t-SNE plot, where you see all the immune subsets that we see in our sample. So there's a large cluster of T cells, that is the lower cluster on the bottom. Then there's the myeloid cluster with a monocyte, which is a cluster on the right. And then NK cells, and on the very top, there's B cells. So I'm now just going to visualize a few of the markers on top of these clusters, which we expect to be seeing from our panel. And the way I'm going to visualize the CytSeq data now is by representing you the same t-SNE plot, but only highlighting the cells that are positive for a certain marker based on the RNA or based on the antibody tags. So CD19, for example, is a very, very prominent marker for B cells. So it should be expressed only on B cells. And when we look into the RNA expression of CD19, we also see the expression, or we only see the expression in the B cell cluster, as we would have expected. And our antibody tags, so the signal coming from our stained antibodies, is also very highly enriched in the B cell cluster. CD11C is a marker for the myeloid population, and we also see it on the mRNA level and also very highly on the protein level. The same is for CD14, which is also a myeloid marker, so it's highly enriched in the myeloid cluster. And as I said before, since these are cord blood mononuclear cells, we also have a small population of less than 1% precursor cells in our sample, and we also pick those up very nicely based on mRNA and also on protein levels. So CD3 is a very well-known marker for T cells, and that's also what we see. So we pick it up on mRNA level, but also on protein level in the T cell cluster. And CD4 and CD8 are also markers for CD4 and CD8 T cells, which should be expressed mutually exclusively in the T cell cluster. This is also what we see. And CD56 is a marker for NK cells, which we also observe in the NK cell cluster. I wanted to shortly highlight two phenomena that are really nicely visible from these data sets here now. So on one hand, if you look into the mRNA expression and the protein expression between, for example, CD11C and CD56, the mRNA has a lot of really salt and pepper-like expression patterns. So only very few cells in the myeloid cluster are positive for CD11C, and very few cells in the NK cell cluster are positive for CD56. Although we know that these markers are really determining markers for these cell types, and they should be expressed in every single cell. And that's an inherent problem with single-cell RNA sequencing, where we have a lot of dropout. So it essentially means the transcript, if we don't measure it, it's probably not expressed, but it's most likely just not detected in these type of assays, which are usually relatively sparse. On the protein level, on the other hand, like every cell that we expect to be CD11C positive, we also pick up CD11C protein levels in these cells. The same is for CD56. And most interestingly, also for CD56, if you look into the NK cell cluster, you really see that there's almost like a gradient of CD56 expression in the protein level. And this is a biological phenomenon. These are the CD56 bright and CD56 dim subsets, which are different subset of CD56 cells. So let me shortly summarize this first part. So we call the method CytSeq for cellular indexing of epitopes and transcriptomes by sequencing. We measure the immunophenotype and the transcriptome of thousands of single cells in parallel. And we essentially don't have any dropout of antibody signals. cell. And we think the method is compatible with any oligo-D-based single-cell RNA sequencing platform. We tested it on DropSeq, in Transcriptomics, and also in the Illumina Bio-Rad DD-seq instrument. So when we are thinking about surface markers, we're used to looking at these biaxial plots from flow cytometry. And here's an example for CD4 and CD8, for example, in the immune system, where you see then this nice mutual exclusive expression pattern between CD4 and CD8 T cells in the immune system. And we can make really essentially the same sort of plots with site-seq data, where you see then the same behavior, the same mutual exclusive expression pattern in the sample, although that's now a completely different experiment. So I simply took this biaxial plot from flow cytometry from the internet. However, what we have in site-seq is we have more information about every single dot here, about every single cell, than simply the protein markers. So we also know the RNA expression and the cluster to which each dot was belonging to from every single cell in this biaxial plot. So what we can then actually do is we can colorize these biaxial plots based on the RNA cluster color. And then what you can see is what you get is something that we call multimodal biaxial plots, which give you really like a high resolution view of which markers are expressed in which immune subsets at which levels. So for example, what you can see, CD4 and CD8 are really expressed the highest in CD4 and CD8 T cells. And then also CD8 is expressed in NK cells, but at the lower levels than in CD8 T cells, but at a high dynamic range or pretty large dynamic range. And also CD4 is expressed on monocytes, but also at lower levels compared to CD4 T cells. So one detail that we still don't know from these type of biaxial plots is where's actually our background? At what point in the data do we stop trusting these antibody counts? And since we have this multimodal readout here in these data sets, so we have RNA and protein data on top of each other, we could simply spike in a foreign cell population as a negative control. And if we see signal on these foreign cells that should be negative for all of the markers, then we know that this signal is actually background, and we could draw computationally then cutoffs to remove this background from all of the other markers. So what we did, we spiked in mouse cells into our immune cell sample, and these mouse cells can be easily distinguished or easily identified from the human cells based on the transcriptome. And if we see signal on the mouse cells, we know that this is actually background signal. So we then plot those mouse cells here on the left plot on top of the human cells, and you see that we have really signal in the mouse cells in a range that is really in the range that we expected the background to be for our human biaxial plot. And then what we can do is we can draw these cutoffs for every individual marker and basically know at what point the data will be still trustworthy and where we are in the background. And this is essentially more direct comparison of background as compared to looking at isotype controls, for example, because here we have really the background signal for every individual DNA barcoded antibody, every individual conjugate in our experiment on those mouse cells. And then, of course, we can make those multimodal biaxial plots or pairs of those multimodal biaxial plots for every marker in our experiment. And then you see profiles and structures that we know very well from flow cytometry. However, flow cytometry, of course, is still the gold standard. So we next wanted to understand is how does our data actually really compare to flow cytometry? And for this, we set out to do two independent experiments. So on one hand, we wanted to see how our data compares qualitatively to flow cytometry. And next, we also wanted to figure out how the data compares quantitatively to flow cytometry. So first, we wanted to understand if we're able to resolve the same proportions of relevant immune subsets in site-seq as in the gold standard flow cytometry. And for this, we basically isolated cord blood mononuclear cells and basically analyzed then one aliquot of those cells by flow cytometry and one aliquot of cells by site-seq using the same markers. And when you look into the flow cytometry, where you basically then enumerate different relevant immune subsets, you can see that, for example, we're able to pick up T cells and B cells by comparing CD3 versus CD19. And then we're able to compare and enumerate CD4 T cells and CD8 T cells by looking into CD4 and CD8 staining. But then we're able also to go down even to the effector cell populations, which are even less than 1% in the CD4 T cells, for example. And if we look at CytSeq data, we see that the data looks very, very comparable compared to flow cytometry. And we're able even to pick up around the same numbers or population numbers as we pick up in flow cytometry. For example, on the far left, we pick up 52% T cells in flow cytometry, and we also pick up 52% T cells in CytSeq. But then when we go to the CD4 cells, to the effector cell populations, which are around 1% in flow cytometry, we also pick up these 1% effector cells in CytSeq. So we next wanted to understand how the dynamic range actually compares between CytSeq and flow cytometry. So for this, we use CD8 as a marker because, as I said before, it's expressed at the high dynamic range in the immune system in different cell types. So what we did, we isolated cord blood mononuclear cells and stained those cord blood mononuclear cells with a mixture of fluorophore-labeled antibodies for flow cytometry and DNA barcoded antibodies for CytSeq, which are recognizing CD8, and then sorted different expression bins of CD8 from very high to very low with a flow cytometer. And then basically reanalyze those different sorted populations once by flow cytometry and once by CytSeq. And when we look into the flow cytometry and compare this to the readout that we got from CytSeq, we see very comparable relative expression patterns between the different bins that we sorted. This nicely demonstrates that the dynamic range between these two technologies is very comparable. To summarize this part, I think it's fair to say that CytSeq data looks very comparable to flow cytometry data. So we're able to pick up the same relevant immune subsets as in flow, but also the dynamic range of markers is very comparable to flow cytometry. However, CytSeq really has the added benefit that we're able to measure the transcriptome of every single cell in our experiment. Now, since we're able to get the protein and the RNA data from every single cell, this should also allow us to get a really much better characterization of every single cell or optimize the clustering in these type of experiments. And as I said before, RNA clustering alone often doesn't resolve cell types that are transcriptionally very similar. So having both data sets on top of each other should significantly improve the clustering. Here I'm showing you an example from a different CytSeq experiment on CBMCs. And I'm showing you the clustering based on the RNA. But I'm already colorizing the cells based on what we know from them from the protein markers. And if you look into the T cells, so the CD4 and CD8 T cells, which are red and green, don't really separate very well in this experiment. And you see the same, for example, for the CD14 and CD16 monocytes. So these cell types are transcriptionally relatively similar, but they have really different roles in the immune system. So now our collaborator, Rahul Satija, is working on ways to integrate both modalities into the clustering, which should really improve the resolution of those experiments. And if you look now into the clustering based on RNA and protein together, you see that, for example, the CD4 and CD8 T cells really resolve into two distinct clusters, and also the CD16 and CD14 monocytes are much greater resolved. So now, as I said in the beginning, when you were looking into our 21,000 cell data sets from synovial fluid, we were only able to resolve 3 distinct T cell subsets or T cell clusters. And even when we look into the original Transcriptomics publication from last year, they were only able to resolve around 5 T cell clusters in their 68,000 cell experiment. And now I'll quickly just give you a quick teaser on our most recent data set, what we're currently working on. So we're currently analyzing a data set of around 70,000 cells with 83 site-seq markers. And we pick these markers to deeply characterize the T cells in the peripheral blood. And having this high-resolution data with these markers on top, we can now really cluster those cells and resolve the T cells at a much higher resolution as compared to RNA sequencing alone. And we really observe 13 distinct T cell subsets in the peripheral blood with our multimodal clustering based on RNA and protein together. So we're able to really to find very clear protein markers, for example, CD4 T cells, which is then divided up into multiple clusters, CD8 T cells, which is divided up into multiple different clusters. Then we see mate cells, we see a distinction between memory naive and even CD4 T regulatory cells. We essentially observe all known T cell subsets in this peripheral blood data set, and we're currently in the process of really analyzing that deeper. So now I'll quickly talk about one site-seq extension that we recently developed, which is essentially making site-seq compatible to the 5' VDJ kit from Transcriptomics, which was recently released and which allows you to essentially make 5' tagged single-cell RNA sequencing libraries versus the traditional 3' tagged single-cell RNA sequencing libraries. So the kit was essentially released to be able to reconstruct T cell and B cell receptors, and is of course therefore really interesting for immunologists, and we therefore wanted to make also site-seq compatible with this kit. This is a quick overview on how the traditional single-cell RNA sequencing library preparation works. So the reverse transcription through oligo-DTs also then contain cell barcode and UMI. So the primer, the RT primer, contains the oligo-DT stretch, the cell barcode, and the UMI, which then results in a sequencing library that is heavily 3' biased, because you have to be able to sequence the cell barcode, the UMI, and the RNA fragment in in one fragment or one molecule. In the 5' kit from Transcriptomics, the molecular biology is essentially just turned around. Instead of having the cell barcode and the UMI on the RT primer, now the cell barcode and the UMI are on the template switch oligo, which then essentially produces a library that contains a 5' or 5' bias, because you have to be able to sequence the cell barcode, the UMI, and in the same fragment also the 5', the region of the mRNA molecule. So the 5' bias libraries have the advantage that you can characterize variable regions at the beginning of the transcript, for example. And that's currently used to reconstruct T cell receptors or B cell receptors, but you can certainly imagine also use this kit to reconstruct other gene families that have variable regions in their 5' ends. Just to remind you, in our traditional site-seq setup, our antibody oligos have a poly-A tail, which is essentially complementary to the oligo-DT primers, which contain the cell barcode and the UMI. And for our 5' assay, we designed the antibody oligos to be reverse complementary to the template switch oligo that has the cell barcode and the UMI appended to it. And if you're interested in exactly how this works, or if you're interested in the sequences, you can go to our website, siteseq.com, where we describe all the details on this protocol. So for proof of principle experiment, we have a collaboration with the lab of Sergei Korolov from NYU, and we essentially observe very similar performance and sensitivity in our 5' site-seq assay as in our traditional 3' setup. So we can really nicely, as you can see here, distinguish CD4 from CD8 T cells, for example. But then, since this is a 5' kit, we can now also, on top of it, reconstruct the paired T cell receptors, for example, in this data set. And here I'm showing you the two most prominent clonotypes from the alpha-beta and gamma-delta T cell receptors. And you can, for example, nicely see that one of the alpha-beta clonotypes is in CD4 T cells, and the other is in the CD8, is a CD8 clone. Since we now have these DNA barcoded antibodies and we can read them in single-cell RNA sequencing experiments, we asked if we can also use those antibodies for sample multiplexing. In flow cytometry or CyTOF, for example, people are using already for a while differentially labeled CD45, for example, to demultiplex or multiplex different experiments in one run. So we asked us, can we do essentially the same thing for single-cell RNA sequencing, and called the method cell hashing. So the way this would work is essentially very similar to flow cytometry. You simply use an antibody or pools of antibodies that are recognizing ubiquitous cell surface markers. So for example, beta-2 microglobulin, CD45 or CD298. And then these antibodies or pools of these antibodies would then be tagged with different barcodes, which we call cell hashtags. And then different samples or only one sample can be stained individually with markers of the different with different barcodes. And then after staining and washing away the unbound antibodies, these samples would be pooled and then passed through one lane on the single-cell RNA sequencing instrument. And then after the run, you could demultiplex the sample based on the DNA barcode that was used for each sample. And one added benefit to that is that you can also identify doublets because you have collisions between different barcodes. Let me now quickly talk a little bit about doublets. So this table here shows the multiplet rate or doublet rate, the increase of the doublet rate in Transcriptomics, the Transcriptomics instrument, and which increases with the increasing number of cells that are loaded. So we typically run experiments of around 5,000 cells, or yielding around 5,000 cells, because this gives you a doublet rate of a little bit less than 5%, which is acceptable for many experiments. Because you don't want to have more doublets because it's very hard to computationally identify these. So the machine is essentially agnostic to how many cells you load into it or how many cells you want to get out of it. So if you, for example, run 25,000 cells, you have a doublet rate of 20%. But of course, that's not acceptable because you have every fifth cell in your data set is going to be a doublet. But having the cell surface marker on top of the cells, so the hashtag on top of the cells, because of the collisions of the different tags, you're actually able to identify the doublets computationally. For example, if you use 3 tags or 3 different tags, you're able to identify 1/3 of the collisions, more or less 1/3. If you use 10 tags, you can identify up to 90% of the doublets based on the cell surface markers. And then what this actually allows you to do, you could superload the single-cell RNA sequencing instrument. For example, in this case, to load, for example, 25,000 cells, out of which 5,000 cells are doublets, but you could identify 90% of those doublets if you use 10 different hashtags. So this superloading workflow would then look like this. If you have a sample that you, for example, want to really get a lot of cells from, you would split it up into multiple vials, for example, 10. And then each vial will be labeled with a hashtag antibody with a different hashtag. And then after washing away unbound antibodies, you would pool those cells, all the different vials back together, and then put those cells all together through one single-cell RNA sequencing workflow. And here I'm basically exemplifying this with 10x. So if you, for example, then pass those cells at very high concentration through one single 10x lane, in order to yield around 25,000 cells. And as I said before, out of those 25,000 cells, there are 20% doublets, but we're able to resolve 90% of those 5,000 doublets because of the 10 hashtags that we're using, which will then end up being only 500 unidentified doublets and around 20,000 single cells, which have then a doublet rate of around 2.5%. For our proof of principle experiment, we wanted to try both in one experiment, the sample multiplexing and the superloading. So we obtained PBMCs from 8 genetically distinct donors and stained each PBMC line with a different hashtag. And also we included some unstained cells as a control. And then after washing away unbound antibodies, we pooled all of those cells together. We then passed those cells at a very high concentration through one single cell 10x lane to yield us around 25,000 cells. And then we analyzed the data first based on the RNA alone, so basically where all the different samples would be essentially on top of each other. But then with the hashtags, we would be able to demultiplex the data and also resolve the doublets. So we first clustered the cells on the RNA, and you see we really recover all the immune relevant subsets that we would expect to see in PBMCs. But then the coloring already gives you the information of the different hashtags of the different samples that we have in our experiments. And you see that they very nicely all fall on top of each other. But then what you can do, you can also cluster the cells based on the hashtags. And then you see very nicely all these 8 distinct islands of numbers of cells that we would expect to see. And very nicely also in between, you see 28 smaller clusters, which are all possible combinations between the 8 different hashtags. It's maybe a little bit nicer to see even in a heat map, where you see that the hashtag signal is very specific only to one subset of cells in the sample. The multiplets are really combinations of all the different hashtags, and the negative cells are also negative. So since we used genetically distinct PBMC donors for this experiment, we could also use the multiplexing method based on the genotype that was recently developed by Jimmy Yee at UCSF and called the MUXLIT in order to test the performance of our method. So here in this plot, I'm showing you the MUXLIT-based classification versus the cell hashing-based classification, and you see these are in very good agreement. So now cell hashing essentially allows you to do multiplex different experiments in one single cell RNA sequencing run. These can be different treatments versus a control, multiple samples that you get, maybe different tissue biopsies, which usually yield only very little samples, and you can now pool those all together to get good numbers of cells. And you can also do experiments which would otherwise be relatively cost prohibitive, where you need to do a lot of different experiments or conditions. So for example, here I'm showing you a titration from flow cytometry, where people check for the best concentration of an antibody, where the signal and the noise actually separates very nicely from each other. And we actually try to do the same sort of experiments for our site-seq antibodies to understand what is the best site-seq antibody concentration that we should be using. And you see very similar titration curves as we also expect to see from, as we also know from flow cytometry. However, overall, the recommended concentration for the antibody from flow cytometry also tracks very nicely with the site-seq concentration that we estimated based on our experiments. So the last thing that we really wanted to figure out is, do actually these antibodies, which have poly-A tails and recognize cell surface markers that are very highly expressed, do they actually compete with oligo-DTs or reverse transcription primers with the mRNAs? Because these markers can be very highly abundant, and you could envision that there's a competition for the mRNA binding sites, for the poly-T binding sites on the beads. So what we did is we essentially used our cell hashing antibodies, which we know are very, very highly expressed in all cell types, which is essentially a pool of beta-2 microglobulin and CD298. And we tested 3 different concentrations of these cell hashing antibodies, and then compared this to a non-hashed control. And then we run 1 lane with the hashed cells and 1 lane on the 10x genomics with the non-hashed cells. And we only ran around 200 cells or so per lane in order to basically, or per hashtag, in order to really be able to sequence those cells very deeply. So here's now the data for the numbers of UNIs and the number of genes between the 2 different between the four different conditions essentially. And I think it's safe to say that there's absolutely no effect on the standard single-cell RNA sequencing output if you have a hashtag in there or if you don't have a hashtag. So with this, I would like to conclude. Site-seq really allows you to measure the immunophenotype and the transcriptome from single cells. We don't see that any dropout of the antibody signal in the cells, and we think that the protein readout is very comparable to flow cytometry, with the added benefit that we have the transcriptome data for every single cell in the experiments. there are easily more markers, or you can easily multiplex more markers in CytSeq as compared to flow cytometry in CyTOF. We've been now going up to over 100 markers easily, and we don't see why there should be a ceiling for this. We think that the method is compatible with any oligo-DT-based single-cell RNA sequencing platform. So we showed it on Dropseq, on Transcriptomics, and on the DDSeq. And we also showed that the method works on the new 5' kit from Transcriptomics. And the multimodal clustering, the clustering based on the protein and on the RNA, really increases the resolution on the characterization of different cell types and cell states. Cell hashing allows you to basically multiplex different single-cell RNA sequencing experiments, but also allows you to superload single-cell RNA sequencing instruments. And we're now basically routinely running 25,000 cells on a 10x lane. And one thing that I haven't really talked about that much is that there's also one more added benefit, that you can also identify low quality or low UMI cells in your data set, because you have this added information value from the cell surface models. I would like to close by thanking all the members of the Technology Innovation Lab, especially Peter, Eleni, and Stephanie. This project was really a great collaboration with the lab of Rahul Satija, who's really just next door to our lab. Christoph and Shiwei did most of the computational analysis presented here. Prateep really helped us to benchmark CytSeq to multicolor flow cytometry. And then I specifically wanted to also thank the people at BioLegend for making CytSeq and cell hashing reagents available. It was really great to interact with Bertrand Young in the last year. If you have questions, comments, or suggestions, don't hesitate to send me an email. You can also follow us on Twitter if you like. And if you're interested in the protocol or in updates to the protocol, we have a website where we keep everything up to date. It's cytseq.com. Thanks for listening in, and I'm really happy to take questions.
Thanks, Marlon, for an excellent presentation. We now have a few words from our sponsor, BioLegend, about their TotalSeq antibodies. The full sequence of the oligonucleotide includes a PCR handle, the 15 base pair barcode, and a poly-A tail. TotalSeq A refers to conjugates developed to work for CytSeq on Illumina sequencers, compatible with the 10x Genomics platform and other poly-A based single-cell feed capture. Other formats may be available in the future, and custom conjugations are welcome. They will put their vast conjugation expertise at your service. Please visit biolegend.com/TotalSeq for the most updated information. Also, BioLegend is committed to supporting innovation with the highest quality standard possible. Their TotalSeq reagents undergo a stringent quality control process that brings together several biochemical and functional assays. First, all lots of TotalSeq products are conjugated to 1 or 2 oligos per antibody. They also guarantee the products contain no detectable traces of unconjugated antibody. Second, all lots are tested by flow cytometry to ensure they stay in the expected cell population and that oligos are attached to the antibodies. This process has been validated by comparison with a traditional antibody-based 2-step flow cytometry staining. The current testing involves hybridization of a poly-DT oligo that is tagged with a fluorophore and thus directly detects the TotalSeq conjugate on the surface of the cell. Third, and finally, every lot is sequenced to confirm the correct oligo and barcode in the conjugate. And now we have a few questions from the audience. If anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. Okay, and so our first question is, let me see if I can figure this out. Okay, so the first question is, how do you deal with your background? It appears that you were showing some mouse controls.
Hi, so so at the beginning for the paper we used mouse as a control because I think that's a more direct comparison as compared to using isotype controls, because you basically see the background for every individual
conjugate that you make directly on these mouse cells, the effect of that conjugate on the mouse cells if you have a really higher background for one specific conjugate. We're using isotype controls now as well, but for other reasons.
The next question is, did you titrate your antibody concentrations in your large panel?
So yes, we did compare. So I showed in one of the slides that we tried to do some like titration experiments for our smaller panel, and then we essentially also did titrations for the larger panel. Although we realized that some of the most of the concentrations that are recommended for flow are essentially the same concentrations that we also need for CiteSeq.
Okay, and then we have a question from Eratz, I'm sorry if I mispronounced your name. They ask, could one cell be labeled with 2 antibodies?
I'm not sure if I really understand the question thoroughly. So yes, I mean, one cell can be labeled with many antibodies. So we're using now, yeah, I mean, we have some panels that go up to 100 antibodies, and many markers label the same cells at the same time. And yes.
Okay, so our next question is from Magdalena, and she asks, can you detect intracellular markers like the FOXP3 using CiteSeq?
Yeah, that's a really interesting question. We're heavily working on getting intracellular staining to work. If you're interested in looking into this a little bit more, there was a recent pretty nice publication by the lab of Klaas Mulder that got intracellular staining to work together with transcriptomics. Although it was a plate-based method, but I think that already goes in the right direction. But we're also working on this.
Great. And then we have a question from SEMA, and they say, let's see, I'm trying to read the question. So how to essentially detect cell subsets. So for example, with cancer stem cell subsets expressing different marker combinations, and each subset with a different differential expression of these markers at transcript and protein levels or both.
Sorry, I didn't really understand the question, to be honest.
Okay, so how do you detect different cell subsets? So they use cancer stem cells as subsets as a example. So you have different cancer stem cell subsets expressing different markers and different combinations of markers. So how would you detect those?
So the prerequisite for this is if you want to detect them based on the surface markers, that you have an antibody that is able to characterize or identify this subset. That's all we, I think, showed. I mean, the easy example for this is the immune system, where there's very nice markers for many different subsets that people have been using for a long time in flow cytometry. If you don't have a surface marker for your subset of interest, then but maybe you have the transcriptome, for example, you can then go back and look into the transcriptome data, and maybe these cells anyways might cluster already differently if you cluster them on the gene expression. But yeah, in order to really identify different subsets, you must be able to, you must have either a marker that you know or the transcriptome.
That makes sense. The next question is from, I'm sorry if I mispronounced her name, is Sarah. And they ask, can you run intracellular staining, including phosphoflow antibodies?
Yeah, that's, as I said, one question earlier, something that we're heavily working on, and I think many people are also interested in that. Right now, I was unfortunately not able to show any updates on this, but yeah, it's a very interesting field for sure.
Okay, and then our next question is from Deepak, and they ask, does CiteSeq analysis require more stringent cell washes than for flow or mass cytometry?
We've actually not really thoroughly tested this yet. So we typically just wash our cells 3 times. I know that many people in flow don't even wash their cells once after staining because they don't really care about too much of a background. But the problem is that all the background that we have is additional sequencing reads that you waste on stuff that you're not really interested in. So we typically just do 3 washes, and yeah, then it works pretty well.
Okay, and then they ask, are the cell losses during the staining process comparable with what, well, you didn't, can you, yeah, they asked if it was comparable with flow cytometry, but you answered that.
Yeah,
Question about not being sure about comparing the two.
I mean, the staining protocol is essentially exactly the same as you would stain cells for flow, and then we just recommend them to wash at least wash the cells 3 times. But probably you can get away with 2 washes as well. We're not really sure.
Okay, that looks like our last question. So that brings us to the end of our presentation. Thank you again, Marlon, for a very illuminating presentation and a great discussion. And thanks also to our sponsor, BioLegend. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the webinar and would like to view the video of the recording of the session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 to 48 hours. There, you can also see the other webinars we have lined up for you on Bitesize Bio. So until next time, good luck in your research and goodbye from all of us at BioLegend and Bitesize Bio.